The nutrient molecules in an unfertilized amphibian egg are dense and are concentrated by gravity in the lower half of the egg, which is called the vegetal hemisphere. At the opposite end is the animal hemisphere. A darkly pigmented cortical cytoplasm lines the animal hemisphere. In amphibians, specific sperm-binding sites are found only in the animal hemisphere, so sperm always enter the egg in that hemisphere. Following fertilization, the dark cortical cytoplasm of the zygote rotates relative to the inner cytoplasm. The rotation of the darkly pigmented cortical cytoplasm leaves a region of diffusely pigmented equatorial cytoplasm on the side opposite sperm entry. Due to its appearance in the microscope, it is known as the gray crescent, and it will be the site of important developmental events. What effect do these cytoplasmic rearrangements have on the development of the embryo, and what is the significance of the gray crescent? Such questions caught the attention of biologist Hans Spemann at the turn of the 20th century. Working with a recently fertilized salamander egg, Spemann used a fine baby hair to split the zygote in half in the plane of the first cleavage, bisecting the gray crescent. When he did this, he found that both halves of the zygote developed into normal embryos. In another experiment, however, one half received the entire gray crescent and the other half received none. In this case, the half containing the entire gray crescent developed normally, while the other half became a clump of unorganized tissue lacking dorsal structures. Spemann dubbed this the Bauchstück, or belly piece. Why should these two experiments give different results? A hint came from fate maps that showed the gray crescent region gives rise to the cells that initiate gastrulation. Spemann and his student, Hilda Mangold, demonstrated that enormous changes in cell potency take place during gastrulation. Using embryos that had just begun to gastrulate, they removed pieces of tissue destined to become nervous system, presumptive neural ectoderm, and transplanted them to a location on another embryo where skin, epidermis, normally develops. The gastrulas were then allowed to develop further. What do you suppose happened in the gastrula when the presumptive neural ectoderm was transplanted to a region destined to become epidermis? Click on one of the embryos above. The transplanted presumptive neural ectoderm developed into epidermis, a tissue appropriate for its new location, indicating that the fate of transplanted cells from early gastrulas is not yet determined. That is, they are uncommitted. However, when the transplants were performed later in gastrulation, Spemann obtained completely different results. The transplanted presumptive neural ectoderm produced a second neural plate in the host embryo, indicating that sometime during gastrulation, the fate of the cells had become determined. The cells became restricted to their eventual paths of differentiation. Gastrulation in frogs begins when cells in the gray crescent, called bottle cells, because of their shape, move inward to form a fold called the dorsal lip. In what would become their most famous experiment, Spemann and Mangold next tried transplanting the dorsal lip from one gastrula to another. They transplanted the dorsal lip onto an area destined to become the organism's ventral epidermis. The transplant stimulated gastrulation at the new site. A second invagination formed, and gastrulation proceeded at both sites. What do you think happened to the gastrula after the second invagination began? Click on one of the choices above. Spemann and Mangold named the dorsal lip cells the primary embryonic organizer because they could induce the host's tissues to change their fates and become determined. These cells organized host and donor tissues into a complete secondary embryo. Since its discovery, researchers have studied this region intensively to uncover the molecular basis of its action.